the condensation, we just go through this definition. Okay, as we are moving toward the complex compound, when one dipeptide and one amino acid will come together, a tripeptide will form. When two dipeptide will come close together, a tetrapeptide will form. When a tetrapeptide and tripeptide will come close together, a heptapeptide will form. Two tetrapeptide will form octapeptide. Like that, we can go for any series. Any natural compound we can select, and then we can hypothetically break down in the laboratory, and we can assemble it through the synthesis. Lower, there is a formation of tetrapeptide is shown, and in the yellow base, there is a tripeptide group. Peptide, I have never coined the term peptide. Peptide is known since 1920 when the insulin was introduced in the clinic. As you are seeing here, several synthetic derivatives as well as the other compounds came up to 1984. In the laboratory, we are dealing with small amino acids. Everybody is aware of glycine, phenylalanine, leucine. But if you go to the literature survey, you will find the derivatized amino acids like isoserine, dihydrohomoalanine. So it is through literature, and even halogenated amino acids are seen through isoleucine. This is an example of a cyclic peptide molecule, cyclolystin A, where you can see the close isoleucine marked by the red arrow, as well as the formyl leucine moiety. N-methylation, this is very important reaction in terms of designing new project. Any cyclic peptide can be synthesized in its N-methylated form, and as literature says, N-methylation modulates biological, structural, and pharmacokinetic property of peptide. So it can be a novel idea. Any existing peptide we can dissociate, we can N-methylate and reassemble it, and it can form new project. So, and it is not like that. So how several projects can be designed? Like we can N-methylate it, it will be a new compound. We can N-methylate here. So within a single structure, there are many variations. And then we can design 5 derivatives, we can compare the SAR, and with natural compound, we can compare, and we can develop the structural activity relationship, which compound is better, and which functional group modify. And likewise, you can do halogenation. That can be another idea to design the fluid. This is N-methylalanine. Usually NH is there is NCH3. But this is natural compound. Okay, through the slides you will send the publication list at the bottom, because all what I completed MCOM in 2001. From that time I synthesized more than 70 peptides. So all are published papers, you will get the citation down. So you can go there and go for it. Just by here I will be able to give you only a brief idea. So here are the example and the product which is mentioned in color, that was synthesized in our laboratory and got published. It will be common in all slides. So like polyheptapeptide A, which is isolated from insect pathogenic fungus, and it is N-methylated peptide, and it was synthesized and got published. First, I am going through a review of literature, and then from the mid we will start how in laboratory we can perform the experiment. So it will give you a thorough idea. No need to go in the library to open the books. Here you take the idea and then start laboratory work. Acidic moieties can also be part of the peptide chains, like you are seeing here a linear peptide with ADPA and AHPEA moieties. Heterocyclic moieties you might have studied in the organic chemistry, thiazole, oxazole, but here those are part of the ring, part of a cyclic structure where peptide bonds are there. So you can see the thiazole moiety, then methyl oxazole moiety, oxazoline moiety, and methyl oxazoline moiety. So these can be part of the cyclic structure which are bioactive. Natural heterocyclic amino acids are also there, like you can see histidine and tryptophan. In histidine, you can see the presence of imidazole moiety, and here indole moiety is present. And these can also be part of the natural structures. So lower you can see proline moiety is there, then histidine moiety, and then there is the indole moiety. And that one are already synthesized by us and got published. This was an interesting collaboration with the University, Wuhan University, China, where we have worked on the natural proline-rich cyclopeptides. And we have seen the interesting stereochemistry there with the cis and trans. Somewhere cis were present at 1,3 position, 1,4 position, or cis or trans were in a particular geometry, and they have shown the biological effect. And this is another parameter in addition to N-methylation, which we can use in our design project. We can say a heptapeptide we design, and there that geometry we can replace, so that a natural compound with that established compound who has shown activity that can be copied, and same bioactivity we can expect for our design project. So here I have separated those compounds with specific geometries. It may not be visible, but you can go through the publication, so you will get a better idea. It is free publication on PubMed. Other moieties like halogenated amino acids, specifically bromo and hydroxytryptophan, these can be part of structures. Okay, this is interesting slide. Peptides can be linear, cyclic, or a mixture of both. Means cyclic with a tail, with a linear tail, or maybe bicyclic. All are found, and all are bioactive. Like these first three peptides are linear chains. It is a cyclic and a linear chain combination. Then this is a total cyclic compound, gramicidin S, a established peptide antibiotic. And here is moroidin, which is an anti-cancer compound with a bicyclic structure. So they appear complex, but if you will understand individual amino acid stereochemistry and geometry on the paper, then this will look very simple. For me, it is very simple, that because I will just focus on one particular part and then I will go through the whole structure. Sources: marine sponges, marine mollusks, they are very rich in cyclic peptides. So here is example of one cyclic peptide from Hymeni acidose species. It was Hymeni amide E, and it got published in itspharmacycollege. It was my M homework during 1999 to 2001. Other sources can be cyanobacteria, hypomycetes, fungi, bacteria, plants. Similarly, dianisosupportose, which is a plant. We have synthesized one cyclic peptide dianisine A, and it published in chemical papers. Sorry. Okay, so like up till now I discussed how the N-methylation is helpful and how this second fact was we can include the heterocyclic component inside it, but introduction of the heterocyclic moiety is not so simple. Okay, cyclic peptides can be synthetic and natural. Both have the anti-cancer properties. Either we can select the natural compounds which are isolated from marine sponges plants. So, like how the projects can be designed, either it can be a pharmacognosy based where we can isolate the compound from the natural resources, we can characterize it and biologically evaluate it. Those are published as papers. Or synthetic work you can do. You can take a structure from a already published paper, which is isolated compound, dissociate in the laboratory, and go for the again associating in the form of linear chain, cyclize it, and compare with the natural molecule. And disrupts the outer membrane, and it occurs through dearrangement of cell membrane, increase in permeability of cell envelope, and leakage of cell content, finally cell death. So this is the possible mechanism for the antibacterial effect. Similarly, here in another slide you can show how cyclopeptide binds with vector to form a complex. Interference with dephosphorylation happens, and finally stent formation will lead to stent formation of the bacterial cell wall. This was slide I found in a paper published after the COVID, where cyclopeptide has an antiviral effect and associated mechanism. Target is viral protein receptor binding domain, and there is restructuring of the spike protein. So I don't know the exact example of cyclopeptide as seen in the literature, which was effective in against the SARS virus. Okay, some of my colleagues do small research. So I'm doing the peptide research. Obviously, I will prefer peptide, but I will tell you the reason why I will prefer peptide. Say example of a wonder drug, paracetamol. Everybody knows about paracetamol. Everybody uses it like toffee. It's simple cure. Will not go to doctor, will not go to pharmacy, just will take the Panadol and fever will reduce. But you know how much gastric side effect it is producing. It is a small molecule available everywhere without prescription. So, if a small molecule is doing like that, then what about the other drug? So, we are mostly using the NSAIDs and all have the gastric side effect. We are treating one condition, second, and they are generating second illness. But I also that does not mean we should not take paracetamol. I am just saying we require the novel drug without the side effect. Peptides we can believe why? Because in our body peptide hormones are present. If they are harmful, then we should not allow it. And if we are living, that means I will not say 100% harmless, but they are safe. That's why if they are in the body and we are. Yes. Okay, so that's why we are alive, and peptide hormones are there, like antidiuretic, and we are taking from outside also. So, and that is the solid base why we should go towards the peptide research. But originality is not not like that. There is cost effectiveness also. These are deal by the biotechnology company. These are not the drugs which will be normally available like paracetamol. That time it will take time to reach that stage. But it is up to us how we will start utilizing more production will be there, cost will come low, and similarly we can believe that same thing happened when paracetamol was first introduced, it will be costly. Then it will opted all started producing the cost is just in pennies. Okay, so they are less cytotoxic, they are less harmful, and they are highly selective. So these are the advantages. If we compare cyclic and the linear structures, cyclic structures are always preferred. Reason is inherent flexibility of linear peptide lead to different conformations, which can bind to more than one receptor molecule, resulting in undesirable adverse effects. Cyclization of peptide reduces degree of freedom of each constituent within the ring. And another is cyclic structure is resistant to hydrolysis. You know the lock and key mechanism? If you will go with a bunch of keys to open a lock, we have to try every key. The same thing happens with our biological response. In our drugs, which is three-dimensional, like we are eating in two dimensions, a solid, but in that API is there, When it will go inside our stomach, it is like that. It has some functional group and we have the receptors. Until a specific functional group will not attach to that specific receptor, bioresponse will not come. And there may be undesirable side effect. That's why stereochemistry also plays an important role. So same thing is there. In linear peptide, both ends are free, it is liable to hydrolysis, and so cyclic preferred in that respect. Cyclic peptides in clinical trials, they are over 40 cyclic peptide drugs are currently in clinical use, and one new cyclic peptide drug enters the market every year on an average. Approximate 140 peptide therapeutics are currently being evaluated in clinical trials. And this is slightly old data that may be increased nowadays. We have some established cyclic peptide antibacterial drugs like telavancin, with their specific yield. This drug is available in market with cyclic peptide in nature. Another approved peptide-based drugs are there in the company name. These are some of the antibiotics which are generally prescribed by the physicians. Vacitration, polymyxin B, intravenous, gramicillin S topical cream, vancomycin IV and oral. So these are available. That means there is trust of peptide research, which will be also attested by my next slide, where US market, how investment is going on peptide therapeutic. There is availability of peptide drugs in market, but there are few. So in the graph, you can see how the US peptide therapeutic market size is increasing from 2014 to 2025. This is global peptide therapeutic market and clinical pipeline inside 2026, which says global peptide therapeutic market opportunity US $65 billion, and out of which cancer peptide therapeutic market claims US $22 billion. Okay, this was also what is in books, what is in journals. Now, what we can do in laboratory. Before starting your laboratory experiment, peptides can be synthesized by solid phase, solution phase. I work on solution phase technique. So before starting your experiment, you must go through the SWOT analysis. What are the strengths, what are the weaknesses, what is the opportunity, what are the threats. You must consult your supervisor, co-supervisor, and discuss the project, what you have designed, what will be the final output. Whether it is just for academic degree or we have to extend it for further to use in the society. So there are a lot of concerns are there. Your project should not be a futile effort, because you and your supervisor will be night and day involved in that project. A lot of finance input will go. And at last, although there is facility, if any wrong thing happened, there are journals like Journal of Negative Reports nowadays. So there is hope for that, but be positive, so go for that. Discuss it properly and go ahead. These are the general reactions which we can utilize for the protection of NH2 group. Both group can be used for the protection. And if you want to reverse the reaction, you can use trichlorotic acid, you will get the PNH2 group. Similarly, the free carboxyl group can be protected by using methylation reaction, esterification, with phenyl chloride. And if reverse you want to do, you have to do the alkaline hydrolysis with lithium hydroxide. This COH group will be free. So this is the basic reaction what we utilize throughout the whole chain, preparing the whole chain of the peptide. This is a both protection mechanism. I will not go in detail of these slides. So these occurs in 3 steps. Both the protection using trichloroacetic acid occurs through these 4 steps. These are some of the amino group protecting agents. Although both you will found in the chemistry, but ACMOG and the CBZ, yes, CBZ are also there. CBZ is the old one, ACMOG is costly one. So that's why both is preferred. Coupling agents which can be used in the, to initiate the coupling of both amino and carboxyl group is DCC, most commonly used EDC, DIC, slightly costly. This is a possible coupling mechanism where a highly insoluble dicyclohexyl urea product can be filtered off and you can get your dipeptide easily. One of the problems with the coupling reaction is if the reactions are not maintained as per the protocol, suppose 0 degrees Celsius for 12 hours. So there are chances of formation of a heterocyclic compound instead of a dipeptide, like oxazolone formation. But that is not your desired. When you go for IR spectrum and CONS bands will not appear, you will understand what it forms. So it is pyrazole. To avoid this situation, sometimes either you have to concentrate on the reaction condition seriously, or you can use a racemization suppressing agent like OF and hydroxycinamide or DMAP. These can be utilized. This is the mechanism of formation of activating ester through OF. This slide shows how the coupling happens. A tripeptide, how a tripeptide will form. A dipeptide after proper deprotection can be coupled with the amino acid to form a tripeptide. Two dipeptides can be coupled to form a tetrapeptide. Similarly, tri and tetrapeptide can form a linear heptapeptide. And finally, through these four steps, that linear chain can be cyclized to a cyclic structure. So this cyclic structure is natural in nature. So this was our first step. When we seen a compound in the published literature, that was the compound. But we took on paper, we broke it into 7 amino acid units, and then in the clockwise direction, we coupled all the amino acids to form. And then we will compare the natural and synthetic bioactivity, so how the response comes. This is cyclization of an methylated peptide. Characterization through PEPMAS. Here the basic step is you can draw the structure on the paper and start one cleavage point, and then you can compare your PEPMAS spectrum with them like this. If cleavage happen in serum glycine 385, 484, 631 peaks are observed in the PEPMAS. So this was a laboratory experiment. It is my journey how this much antimicrobial peptides we have synthesized in the laboratory and got published. These are anthelmintic peptides we have synthesized, cytotoxic peptides we have synthesized. Investing we worked on these 6 peptides during 2016 to 24. These are some of our ongoing projects where these are the projects where you can collaborate with our laboratory. So key takeaway are either you can isolate the compound and do a pharmacognosy type of study, or you can go for designing your own compound or keeping the basis of an already published paper on natural peptide. And methylation you can utilize, then the fraction you can introduce, like I shown here. Here, like any established antibiotic is here. You can select, you can go to literature and see what is the common component in all published paper. That component you can introduce in the newly designed compound. And like if you will choose gramicidin as which is antibacterial, so obviously your compound antibacterial effect will be modified. This is the scientific output of our laboratory there. The book chapters, articles, some of the university we collaborated in past years. So we are involved in a lot of community outreach program there. These are some of the programs we did. Every year we go and celebrate these days. This is my laboratory. Some of my colleagues in our this activity. These are some of my achievements. I got the Outstanding Researcher Award, Distinguished Pharmacy Researcher Award from their local organization. Okay, this is very interesting slide just to end up. So we are in India. Yeah? So this dot is Trinidad. This is part of West Indies. It has population of 1.4 billion. That is not increasing much. It is likewise since many years. They enjoy life in different ways. Their street food is doubles, which is like our chole bhature. So it is available at every corner. It is famous for carnival. If you will visit, in fact, there is carnival festival with different. Although all West Indies islands have their own carnival, but Trinidad is famous for. We have beautiful beaches, and we have our beautiful campus of University of West Indies. So thank you very much for listening to me. Any questions?
That was truly an insightful session. Thank you so much, Professor Rajiv, sir, for such a compelling presentation. For chairing this session, we were privileged to have Professor Dr. Anurag Chaudhary, sir, Meerut Institute of Engineering and Technology, Meerut. We are now going to open the floor for questions. So if you have any questions, so please raise your hand so that we'll bring the microphone to you.
Hello, am I audible? Yeah. So first of all, thank you very much, sir, for this lovely presentation. It was very insightful. And I myself, Abu Talab from Jamia Hamdard, is currently pursuing masters in pharmaceutical chemistry. I'm also working on solid-state peptide synthesis. I'm trying to synthesize the temporal peptide. And my question to you is, I run into this problem of cyclization as you mentioned in your presentation. So according to you, what is the best way to avoid that problem when we are synthesizing a peptide?
For cyclization step here.
Yes, sir. Yes, sir.
I showed you the slide for 4 steps.
Yes, sir.
Sometimes we also struggle in that step. We run the reaction multiple times because we keep the linear peptide in the final step for 7 days at a controlled temperature, and sometimes things are not proper. So I will suggest strict observance of the reaction conditions and then stick to the spectra. Don't manipulate.
Yes, sir. I will definitely try to do that. So basically, the crux of your answer is to stick to the protocol and don't try anything else.
Yeah, it's, sometimes we are in a hurry. Okay, it is for my M.Com, it is for my PhD, just complete PhD, I will not take it. No. When you will publish that data, it will be used by the other scientists to cite. That is not a joke. And depending, that is drug discovery. That will lead to the, if you will do the wrong thing, that wrong thing will be cited, and people will scientists will lose a lot of chemical and financial, which is nowadays a big problem. So just go for the true data, what you got, that's all. Degree will be awarded, degree, you work you have done, degree will be there.
Thank you, sir.
ज़्यादा एक तरफ ही रखो क्योंकि जैसे ही आप घुमाते हैं ना फोकस होना बहुत मुश्किल हो जाता है।
सर जी सब हो जाएगा।
नहीं आ रहा
Any other question?
ब्लड आता है।
ब्लर इसलिए आता है कि मैं घुमा रहा हूँ।
So thank you, sir, for answering the question. Now may I invite our director, sir, Dr. S. Sadish, sir, to felicitate our speaker with certificates. Please give a huge round of applause for our director, sir, and Professor Rajiv Raiya, sir. Thank you, sir. Thank you. Now may I invite the chairing person of this session on stage, And I also would like to request Dr. Rajkumari ma'am, Dean at ITS College of Pharmacy, to felicitate our cheering person. Please give a huge round of applause for our cheering person and Dean ma'am. Thank you, ma'am, and thank you, sir. Now, let's move on to our next presentation. And for introducing our next speaker, I would like to invite Ms. Ujjwal ma'am, Assistant Professor of Pharmacology, to please read the profile of our next speaker, Professor Ajay Sharma, sir.
Good morning, everyone. I am Ujjwal Bharti, Assistant Professor of Pharmacy. Would like to welcome you during his studies. His research area includes drug discovery and development from natural sources, immunopharmacology, natural product chemistry, metabolomics, and plant biotechnology. He has published more than 90 research articles in peer-reviewed journals of national and international. He has presented more than 150 papers in national and international conferences. Sir has been contributed 4 books and 6 book chapters. He has published 10 Indian patents and has been awarded with research grants from Ayush, DBT, AICTE and MBCST. Sir has been awarded with Claude Leon Foundation Scholarship in 2013, University of KwaZulu-Natal, Pietermaritzburg Research Center, Plant Growth and Development, South Africa, and postdoctoral research scholarship from College of Pharmacy, International Islamic University, Penang, Malaysia, and Best Teacher Award during Eminent Teacher Award 2019 program organized by MIT University, Madhya Pradesh in conjunction with Dainik Bhaskar Wonder. Sir has been awarded with APP Young Achiever Award during 2-day international conference on current trends in pharmaceutical education research, Edinburgh International College, Viratnagar, Nepal. He has been listed as top 2% scientist as per the list of Stanford University consecutively since last 2 years. Thank you.
Thank you, ma'am, for this concise introduction. Every great discovery begins with a thought. Let your thoughts be heard. Let your knowledge be inspired. Let your words make an impact. With this positive saying, I request Professor Ajay Sharma sir to please start this insightful session. And for chairing this session, we have Professor Dr. Nitin Sharma sir, Center Head of Pharmaceutics Department at Amity Institute of Pharmacy, Amity University, Noida. I would like to request Professor Ajay Sharma sir to please come up on stage and give an initiate to the presentation. Please give a huge round of applause for Professor Dr. Ajay Sharma sir.
That marine peptides, the pharmacognosy you people know about it, the analytical chemistry, biology as well, the researchers, the faculty members, directly or indirectly they are basically taking care of medicinal plants or the individual phytoconstituents for their research work. So if you see the core or the base, it is somewhere around the natural products. And you see the demand of the natural product is very high. But what we are lacking is the availability of the standardized raw material. And that's why we are far behind than the Chinese system of medicine. because of lack of standardized, plant materials, the lack of availability of developed monographs, of course, as well, for the plant materials. That's why this topic is very, very important in the current context when we are talking about drug discovery and development. They were well aware about this important, concept. They were well aware about this important, concept, the drug discovery and development. That time, that much technology, that much, scientific, the facilities were not available, but the Vaidhyas or the persons who were involving in this particular area, they were well aware about the drugs, how to convert it in particular formulation and how to administer it. So I will be touching upon some of the important context of revolution of herbal drug technology. So you see what happened during the first revolution. Whenever we are talking about the herbal drugs, always we have to refer our ancient books, our Bibles, and most of them are basically mentioned in Ayurveda. You know the name of Charak Samhita, Shushruta Samhita, but you see the first revolution which happened in the herbal medicine. So this particular area is not new. You see or remember the episodes of Ramayana when the Lakshmanji was hit by Meghna and how he got hit because that time the Hanumanji went to Lanka and then he brought that Sushin went and they administered Sanjeevani beauty. So that time the peoples were using the herbal medicine, and of course this is the first revolution which has happened in the area of herbal drug technology. So that time there were n number of textbooks which we are popularly calling Ved. They were available and there the information with respect to herbal drugs are mentioned. There were n number of scripts, n number of herbariums were available which are coming under the first revolution, and the concept of making herbarium, which is very, very important while working in this particular area, and you see the concept of immunopharmacology, which is now very, very important. So whatever the traditional themes are there related to the herbal medicine, now we are scientifically proving them. So these all are, the developments or the episodes which happened during the first revolution and second revolution respect to the herbal drug technology. But that time there was no existence of drug and cosmetic acts. Now this is the third revolution, the concept of the preparation of herbal extract scheme. So now you are also preparing herbal extract in the laboratory. Tools and techniques we are using for making it, and the concept of the electron microscopy came, the isolation and the characterization of phytoconstituent, high throughput screening, the concept of leads and the hits, that is computer-aided drug designing and molecular modeling came in existence. So all of these things are happening during the third revolution of herbal drugs and technology. Now you see the fourth revolution, which is important, the concept of in vitro biotechnological tools, the concept of genomics, metabolomics, and the proteomics came in existence. The concept of biotransformation also came. The use and the application of electron micrographs or electron microscopes for various purposes. The laser confocal microscopy is still there, and there are a number of other techniques, including the genomics, came during the fourth revolution. So you can see how the development has been taken place in the area of herbal drug technology, which is directly related to the drug discovery and development from the natural sources. Now, this is the post-COVID era. We all were struggling. The whole world was struggling during the COVID times, and n number of hit and tries happened during the COVID times, and unfortunately we could not got any particular medicine to cure or to manage these symptoms of COVID-19. And during the COVID time, all were using the herbal drugs in one or another ways. We were using whatever the people are suggesting, you use ginger, you use clove, you use ashwagandha, you use turmeric. So by one or another way, knowingly or unknowingly, without knowing the facts and figure about those particular herbal medicine we use, the herbal drugs to combat the effect of COVID-19 virus. And the preparation of that, different formulations came to the market. We were using n number of antiviral drugs, and later on we come to know these are totally non-effective against the COVID-19 virus. After बहुत सारे examples that time you have seen how the peoples were struggling during the COVID times. So our university, we took this particular challenge to come up with the herbal drugs and the products which they combat the symptoms and the effects of COVID-19 virus. And during the COVID-19, as during the introductory lecture, Mr. Zhu told about how the drug discovery has been taken place in our university. So we have developed this particular product against the COVID-19 virus, that is chloroquine. But we tried during this journey with respect to the Ayush permission. We did not get, because earlier we were launching this particular product as a drug to the market, but we did not get approval from the Ayush ministry. Later on, our team decided to launch this particular chloroquine to the market as a form of nutraceuticals or the food products. So the product is now in the market in India as well as in another country. So we have transferred or shared this particular technology to one of the company which are running their plant in US and Missouri as well as in Chennai. So like this, we are dealing with the marketing strategies. Now the 21st century where we are running now, there are n number of techniques and the terms which are very, very popular, and we are hoping they will provide the solution of one or another problems. Now you see the importance of the biotechnology, herbal drug technology, and the information technology. In the earlier lectures, the experts, they talk about the use and the applications of artificial intelligence. So you see the revolution is going with the use of these technology in one or another way. Now in pharmacology or in the herbal drug technology, we are using different terms in one or another way. The concept of phytopharmaceutical came. So this is related with the herbal drug or the extract which are having at least minimum 4 bioactive principles. And we are basically come across another important terms like nutraceuticals, biopesticides, cosmeceuticals. So you see the importance of herbal drugs, how they are becoming popular and the people are working on it. And you see the over here, this particular figure, around 88% of the world population, they are based on plant-based medicine. That's why this particular area is very, very important, and we have to take this particular area of research as a challenge because the lack of documentation and scientific validation. Now you see the Indian system of medicine, it is not new, it is very old, and we all are aware about the various concepts, the Ayurveda, Siddha and the Unani, and the drugs or the products which are mentioned in the system of medicine, they are easily accessible, affordable and effective. That's why they are one of the choice, for the global community as well. You see the herbal drug market, you can see the market capital of Patanjali nowadays. So this is not only just for usage purpose. N number of business potential is also associated with this particular area. So the young people and the researchers, they can make their career and they can go for entrepreneurship in this particular area of drug development and research. And as per WHO, by the year of 2050, it would be around 5 trillion US dollars. So business potential is associated with this particular area. You can think about that. And you can see the Indian scenario of the herbal drugs. Our area is around 2.4% of the world. We are rich with biodiversities and we are having 30,000 plus